start. Okay, so thank you for coming to our first UlyssesNeuro Talk. It's a pleasure to have you here. Now, the first thing, I think the first great challenge that we have been able to achieve and then to do actually is that we are actually live stream at the moment on YouTube. So I want to say thank you to Carolina. She did a great job. Thank you to Mo, who is in UK at the moment. He's our social marketing guy. So Mo, if you can hear me, thank you very much. He's in the air. And we have Ali is there and is trying to as well manage something with a live stream. I don't know what they've done, but we are live stream. So we'll see. Okay, so again, welcome everybody. So this is our first UlyssesNeuro Talk of the year. So our guest speaker is from Italy, it's Alessandro Gozzi. Since this is our first UlyssesNeuro Talk, our idea is that we give you also a brief overview about Ulysses Neuroscience, who we are, what we do, and how we want to fulfill our mission. We are a new entity in Trinity College, so I just have a few slides on that. After that, there will be John Keeley, the senior scientist at Ulysses Neuroscience, that will present our new project on Fragile X in collaboration with Future Neuro and Matthew Campbell. And then we will have Alessandro Gozzi talking about MRI applied to neurodevelopmental disorders. So just an introduction about Alessandro. I don't probably need to do a lot of introduction for Alessandro because it's very well known in the field. So Alessandro is from the Italian Institute of Technology, which is based in the University of Rovereto, and is leading the MRI group in there. I know Alessandro since before the new millennium. So we started to work we met in GSK when GSK GlaxoSmithKline had a big research center in the southern of Europe, and this is Verona, which is our hometown, and is one of the things that we share. And we actually met in GSK. We were working in different groups, but at a certain point, at that time, I started my career by doing voltammetry. So I was spending essentially hours and hours and hours implanting carbon fiber biosensors in the brain of rats and mice. And then at a certain point, they decided that they had to refurbish, I think, the labs. And so they didn't know where to put the voltammetry team, which was made of 2 people, essentially. So there was not a lot of space for us, but at that very time, they also had a new MRI unit, which was led by Angelo Biffone. And you, Alessandro, you were moved from the behavioral work to work in the MRI at that time. And they found a little space for us in the basement where the MRI was, because there was one part that you didn't use. And they said, okay, let's put the voltammetry in there, those guys in there. And so this is where we met with Alessandro, and this is also where Alessandro started his career in MRI, working with Angelo Biffone. And then the other thing that we have in common and that we share, and this is, I think, is a message for everybody, especially for the students or the young people, it's never late to change your mind and start a career. So we both started our PhD quite late. So I was 30 years old when I started my PhD, and Alessandro was 35. So of course now we are very young. So it was just 3 years ago that we started our PhD essentially. And okay, so then Alessandro was awarded a very prestigious ERC grant, starting grant, to set up his lab, which made already an amazing and great impact in using fMRI in rodents, especially in mice. And he has already given a great contribution for anything that is related to X-linked brain disorders and neurodevelopmental disorders, in particular on Fragile X, autism, some aspects of autism, and CDKL5 deficiency disorder. So that's the introduction on Alessandro, and we are looking forward to his talk. Now, a few things about us. So what is Ulysses Neuroscience? Well, we are a private R&D organization, and we are very new. So Ulysses Neuroscience was launched in June 2019. And, I had the idea, this crazy idea, to open my own company after having experience in the industry, having experience in the academia, as you know, having experience in contract research organization. So I thought, okay, we have to do something new. Maybe we can try to put everything together and to have something new and innovative. And this is what we are trying to do in Ulysses Neuroscience. So our identity is that of a private R&D organization. Indeed, as you can see from these bullet points, which I'm going to read together with you. So we are on a mission to operate on a new business model that can make a difference to current practices in order to begin a new journey against brain disorder. So we have our own scientific idea. So we are driven by an hypothesis. And this hypothesis is that we do believe that to advance drug discovery, nowadays we need to have an approach called back translation, meaning that we start our work with the humans, with the disease populations. We try to identify biomarkers of disease progression, biomarkers of pharmacological activity as well, and identify targets in humans. Only when this is done, then we move into the animal model. And if we replicate in the animal model what we have found in the humans, then it means that we have the right model to test our drugs. And this will mean more rapid drug discovery projects, less waste of money from pharma company, which hopefully will all go back to neurosciences to change the approach to drug discovery. The other mission that we have, and I really, I mean, it's really, I really care about it, is that that new journey that we want to take is really something that has to benefit not us as individuals, as researchers. I mean, it's not only for having a nice Nature paper, it's really to benefit the patients. And so something else that we have in Ulysses Neuroscience is that the patients and their families are really at the center of our work. We meet them, we talk with them, we learn a lot from them, and it's really spectacular, you know, what the way in which we can drive our research by talking with them, by knowing exactly what they feel to be the symptoms that are mainly affecting their kids, and what are the drugs that are working, the drugs that are not working, and the drugs that have actually side effects. So we also want to work as a sort of bridge, putting together the academia, and we work with the academia, and John will tell you about our Proteus project with Future Neuro and Matthew Campbell. And we also work with funding agencies. We do apply for grants, and there are grants from the European communities, but also other funding bodies which are dedicated to small, medium enterprises. We do this by ourselves or together with academic partners. And we do work with pharmaceutical companies, which are our main source of income. Okay, so how do we organize our research? So we have a strong in-house research projects going on at the moment, and this is the difference. This is why we call ourselves a private R&D organization. This is our foundation. We do our research, we go to talk with pharma companies. If they like our research and they see that our research can be of help for the drug discovery project, then they collaborate with us. It's not the other way around. It's not the pharma companies coming to us and telling us what to do for them. It's different. So we have organized our work in 3 different research platforms, which we call RP. So we have an RP1, which is dedicated to neuropsychiatry, neurodevelopmental disorders. We work on depression. We work on anorexia nervosa. We have a wonderful project on anorexia nervosa, which is the first of this kind in Ireland, I believe. So we have applied with the ethical committee for this, has been accepted. We're going to start very, very soon to work with patients. We have a project on Fragile X syndrome. For the moment, it's a preclinical project. John will tell you more about this, but we're also going to work with patients. And we have a project on CDKL5 deficiency disorder, which is another X-linked brain disorder. We do this mainly with my academic team, but Ulysses is also going into that. And we do work with both the patients, which is great, and also with animals. The research platform 2, RP2, is dedicated to neurodegeneration, neuroprotection, and neurorepair. We work on aging, we work on Alzheimer's disease, we work on Parkinson's disease. We have some very good projects on that. And we are starting something that you will hear in the next UlyssesNeuro Talk, the 28th of February. I think Charlotte will announce that, which is a neurodegenerative disorder. It's a disorder of the peripheral nervous system, which is called Charcot-Marie-Tooth disease. And so we are going to start both a clinical and a preclinical project on that, and we are very excited about it because it's really new. The third research platform is the platform dedicated to synaptic plasticity and microtubule dynamics, and this mainly analysis of peripheral biomarkers is a key, and is at the service of the other 2 platforms. So we do analyze biomarkers with this platform in both neurodegeneration and neurodegenerative disorders and neuropsychiatric and neurodevelopmental disorders. I talk about back translation, so we do also partner with different companies, and I want to mention one of our key partners, which is Brainwave Bank. We have just signed a partnership, I think in November, just before the SFN in Chicago. So we do work together. We go and see pharmaceutical companies together. And what they do is they have a fantastic wireless device for doing EEG, which is coupled with a tablet where there are cognitive tasks. Now, what we try to do with Brainwave Bank, and the reason why we decided to get together, is because we want to back translate the data that we're gathering, and we just finished our first study with healthy volunteers treated with ketamine in the University of Magdeburg. And what we want to do now is to back translate what we have done in humans into the rodents, trying to do more or less exactly the same. And this is my fantastic team, and what you can see in this picture, guys that have really contributed to the beginning of Ulysses. There is a great motivation. I have to thank my team for the help. I mean, 7 months and we are doing really, really well. And I think there will be some announcement from Charlotte, which is principal scientist at Ulysses now. I don't know. Yes. So wait a minute.
Okay. Thanks very much, Max. So I just want to make a couple of quick announcements. We'd like to take this opportunity to highlight the Ulysses Neuroscience Minerva Travel Award. So this award is open to all young researchers traveling to Glasgow during the summer to present their research at FENS. And the application deadline is the 29th of February. So for anybody interested, please do apply, because it's a really nice opportunity to get some funding towards those travel expenses. And there'll also be some potential new positions opening up within Ulysses in the coming months. So keep an eye out for those as well online for anyone who's interested or might have students who would be interested in that as well. And the other thing I would like to mention is just that our second UlyssesNeuro Talk is planned for February 28th, and this is with Dr. Sinead Murphy, who is a consultant neurologist in Tally University Hospital, and she's going to talk to us about her research in Charcot-Marie-Tooth. So if you're available, please come along, or it should be live streamed as well, so you can also check it out online. I think that's it. I'm going to hand you over to John, who's going to talk about the research project at Fragile X. Thanks very much. Yeah, let me do this.
Hi all, I'm just going to switch this and I'm going to not spend long so we can get on to Alessandro's talk. So I'm going to talk to you briefly about a new project we're just starting with Matthew Campbell's lab and Future Neuro, and it's called Proteus, which sounds very exciting. So we'll be looking at synaptic plasticity, microtubule dynamics, and neurovascular processes in Fragile X syndrome. So we'll be working with Matt and specifically as well with Chris, who is in the audience there. And Matt, I did my postdoc, one of my postdocs with Matt, and he has an amazing expertise in blood-brain barrier and neurovascular biology in the brain. So we'll be working with him to kind of share our expertise in synaptic plasticity, microtubule dynamics, and the blood-brain barrier in neurodevelopmental disorders. Why Proteus? That's Proteus there. He's known as the Old Man of the Sea. He's the son of Poseidon, as we all know and are very familiar with, I'm sure. We all have a big doorstop of classics beside our bed. But what's interesting about him is he himself is plastic. He's a shape-shifting god. And he's very difficult to pin down because he keeps changing, and he is able to change. In relation to our own name, he is in the Odyssey, and one of the chapters in James Joyce's Ulysses is referred to as the Proteus chapter, where he basically mangles language through different languages, stream of consciousness, and it's a very plastic thing. So it's a nice metaphor for what we're looking at, because both, obviously, neurons, we're familiar with them being very plastic, but the neurovascular system in the brain is also quite plastic. So we've established the first colony of fMRI knockout mice in Ireland here at Trinity College. So there's a classic kind of rodent model of Fragile X syndrome. And so what we intend to do in this project is to look at microtubule dynamics across neurodevelopment in these mice, using the LICOR Western blot system, Western blot system. And we are going to look at blood-brain barrier permeability and in these mice. So there's an image from Chris's paper in schizophrenia. We'll then look at tying these to each other, looking at the relationships between changes in microtubule dynamics and blood-brain barrier and how this affects brain function in Fragile X. And then we'll move it into behavioral and seizure activity in these mice to look at their contribution to pathogenesis. So the long-term plan as well. So we know that in Fragile X syndrome, in patients with Fragile X syndrome, that there's been changes in functional connectivity described in these patients. And we also know that we can use functional connectivity as a translational tool in rodents and humans. So we're going to be working with Professor Alessandro Gozzi to develop this here in Trinity College. And allow us to use it as a biomarker in to look at drug deficiency, drug efficacy, drug discovery in fMRI mice and other neurodevelopmental disorders. So I'm going to stop there because I'm the wrong person to talk about this right now. And the right person to talk about this is Professor Alessandro Gozzi, who's now going to come up and chat to you about his research. I presume it's more than a chat, rather a bit more formal than that. Okay, so thank you.
Thank you. Okay, can you hear me? Can you hear me well? Okay, so I'm just taking this because I need to time myself and I want to see people falling asleep. So I'll try to keep this within the my seminar within like 25-30 minutes that there was you know, initially allowed to, but you know, let me know if we need just to shorten this. And well, you know, and of course I have to start by thanking Max and you guys for coming up today. Max for inviting me here. I was really, well, it was the first time in Dublin, so I was really, you know, blown away, beautiful city. And I was also very struck by, you know, the level of organization of Ulysses, a new company, which provided me also with the title of my talk. So it's the first time that I'm invited to deliver a talk, and I'm given a title, and I'm very happy with this actually. So this is the title that they provided with it, and you know, all the keywords are you know, properly emphasized. There's also this repetition, but also just to make sure that we're not talking about that. I'm gonna do that. So very happy to give you like an introduction about what you can do with fMRI, the translational tool. I, however, took the liberty to add a bit of a subtitle, so to speak, to keep some of this research I will show you within like, the context of translation and the context of neurodevelopmental disorders. So in the second part of my talk, I would like to touch upon this point here, this method, which is how to use this translational readout to try and pass the heterogeneity of our autism neurodevelopmental disorders. Yeah. All right. Okay, so that's where I come from. Well, that's where my lab is, as Max said. Yeah, we both grew up in Verona. My lab is in this place here. I always put this slide in because Rovereto, so where we are, it's a bit of an obscure place. So it's a very nice place. So if you want to avoid the start of the place in the train from Verona, where the lake is. My lab is called Functional Neuroimaging Lab at the Italian Institute of Technology, and much of our research deals with a better understanding of the functional organization of the mammalian brain at the macro scale. So we do system neuroscience. We are interested in understanding how the brain works when you look at it like if you were a machine, right? So I call a brain software approach. And as I mentioned, a lot of our research gravitates around this kind of thing. So we use a lot of MRI, not only MRI, but a lot of it. And this is for two main reasons. The first is that, as Max mentioned, I was trained as a translational neuroscientist, so to speak. So I kept this interest in trying to address questions that are directly translatable across species. And I try to make this point as strong as I can in my talk, okay? So fMRI is one of the very few methods, also, rooted in biophysical measurements that can be really directly translated across species. Of course, we have caveats. Mice are not humans, but I try to touch upon this during my talk. And the second thing is that fMRI is really ideally suited for studying what we are interested in, that is, the functional organization of the brain at the macro scale. So how so? Well, this is a concept that many of you are familiar with, but let me just try to introduce it well and properly such that you can follow through my talk. So fMRI is, well, a lot of research that was carried out in humans showed that fMRI in the rat skin brain is an excellent readout for understanding, for investigating the functional architecture of brain function. How? Well, as mentioned, maybe you know already, but so what was discovered pretty much 25 years ago is that if you lie as a volunteer in an fMRI scanner, which is, by the way, the same scanner you use for your knee or whatever problem you can have, and you're not subjected to any task, these spontaneous fluctuations in fMRI signal are not following some sort of a random noise pattern, but are in fact exhibiting a lot of spatial and structural and temporal structures. So they are highly structured, both in time and in space. What does this mean? Well, this means if you look at the spatial structure, it means that these fluctuations can define networks. How? Well, this is the concept of functional connectivity. It's very, I think, effectively illustrated here. So if you just take two brain regions, the prefrontal cortex, posterior cortex, so to speak, you can see that these fluctuations are in fact correlated, right? When you have peaks here, you have peaks here, and the other way around. And this correlation, like in EEG and other methods, is considered to be like an index of functional synchronization. And so you somewhat imply that these two regions must be connected. So hence the concept of functional connectivity. As mentioned, these fluctuations have a lot of structures in space, which means that you can get networks, so regions that are coherent. Okay. At the whole brain level, you can get what is called like a functional fingerprint or functional connectome, which is many considered to be like specific of an individual. So my functional connectome might be a different from Max's and everyone else's here. And of course, it is affected by development, by aging, by memory, and so on and so forth. So here is where the interest in this method lies. Okay. And the reason why we got interested in this approach is that, probably the most important reason is that these mechanisms, the activities within the fMRI, as well as the method and functional connectivity, are almost invariably disrupted in any brain disorders. Okay? And of course, this is very prominent, well, not exclusively, but very prominent in schizophrenia and autism, or all these developmental disorders. So these are the facts are also obvious, right? Because you have this deviant trajectory of wiring and so on and so forth. However, despite a lot of research, a lot of money being poured into initiatives to map the brain and using this functional connectome, both in health and disease, we are stranded with these major fundamental questions. One of these is, but there are many, is what drives this functional synchronization? What are the fundamental elements that are necessary or sufficient to make this phenomenon? The second big question, which is very important for me, are there shared pathological motives that lead to this aberrant functional connectivity? In other words, can we reverse engineer these signals into biophysical events or physiological events that can help us treat or diagnose brain disorders? So that's what we'll be striving to do. And our recipe or our proposition in order to tackle these very ambitious questions is depicted here. So my take here is that if we have trouble in understanding what this means, is because we've been not very efficient in translating models of brain function across levels of inquiry. What does it mean? Well, it means that we are now pretty good at modeling brain function at a micro scale. We can map effectively brain-wide patterns of functional connectivity, but we don't know how to go from here to here. And so what we propose, as Max said already, is perhaps to go, to try and narrow this bridge by going backwards, by trying to back translate this into model organisms, whereby, in which basically we can introduce causal perturbational methods that allow to disambiguate these correlational measurements, because ultimately functional connectivity is a correlation across signals, and if you want to disambiguate, you have to perturb, whether genetically with genetic models or opto, chemo, whatever you like. And so towards this goal, to this aim, we started this program a few years back. And what I'm showing you here is the first demonstration that in the mouse, we can effectively, on the right, we can effectively map these resting state networks. So, and this is interesting because we've effectively started a field of research now that is pursued and followed by many labs across the world. And that's this is really interesting in biology, my friend, the translational meaning of this. Why? Because, as mentioned, you cannot translate the same measures across species, right? And one of the things that is appearing, for example, here, is that some of the fundamental organizational principles of fMRI networks in rodents are, in fact, very well conserved in higher species, such as primates and humans. One of these is reported here. These are networks in the mouse. As you can see, they're all pretty much bilateral, so symmetric. Homotopic is a technical word. And you can see this is just a defined as well. But not so exciting. What is more exciting, however, is that in rodents, in the way we also show that there are these networks that are evolutionary precursors of key important networks of the living brain, such as a Selins network, such as a deformable network. Why am I bringing this up? Because these networks are very often impaired or aberrant in brain disorders. And of course, these are ancient networks because they impinge on the like visceral or limbic cortices that are present in rodents. Obviously, keep that in mind. We can, of course, translate this insofar these networks that we are trying to tackle or to perturbate are evolutionarily conserved, right? There is obviously no, language network in rodents. And then we found a number of subtle correspondence. Probably these are too technical for this talk. Now, before I show you how we can apply this to autism and what we've been doing, let me just touch upon another important concept of characteristic and educational concept for those of you that are in the field. Now, one question that we are trying to address in our lab, what's the meaning of this? And I want to try to educate some of you, especially for those of you who are in the field of imaging. Okay, so sorry if this sounds like very basic, there are some images here. But one question is, okay, well, this is a very important question. Let's start by saying that there is no consensus about what this means. Okay, there is a lot of research that is needed, and there's a lot of controversy. But one way to tackle this problem, both in humans and in mice, is, for example, that's the concept of functional connectivity explained before, right? So two regions are synchronous because we assume that these two regions are connected. So one way to understand what this means is by comparing, for example, either in humans or in mouse or the species you like, functional connectivity, the structure, with a structure of structural connectivity, like the axonal connectivity, which we know very well. I'm sure many of you know that the mouse connectome was published a few years back by the Allen Institute, so it is available. And then also offers me the opportunity to introduce a couple of slides, an unpublished work, where we try exactly to do this, right? To see how the topology, the architecture of functional connectivity correlates with underlying structure, okay? However, what we did in this work here, instead of using the classic, parcellation, you know what I mean, like regionalization of this connectome that has been released, we wanted to do it with no constraints, just voxel by voxel, because in 3D in imaging, we call them instead of pixels, voxels. Because the idea is that, when you use parcellations, you assume that all regions are homogeneous, you have all the voxels, And there are a number of problems. So we want to do it with very, very high resolution. This is the sort of result you can get. Now, this is getting a little technical, so I'll try to get to the point here. I want it to be shortcut like this. But just to show that this level of resolution, this is the axonal connecting mouse. When you come up here are regions that are called hub regions. So very important in integrating signals. And perhaps not surprisingly, you can see we have the frontal cortex, dorsal campus, even the amygdala, now we have enough resolution to see, and so on and so forth. So this is just an example. But why is this interesting? This is interesting for the reason I'm showing you here. So one thing you can do with the connectome, with external connectome, is to find communities. So all these nodes that are bound together, that are tightly linked together, okay? To define module talk communities. And we can use some algorithms that are important. And these are some of the macro scale communities that we found in the axonal connectome of the mouse. And what I find really striking is this. When you compare the topology, so the distribution of this with the distribution of the corresponding functional connectivity communities, FC stands for functional connectivity, and this is not the only institute, this is data in my lab, you can see that they're pretty much the same. They are overlapping, as much as you would hope so. It's interesting, right? Because this suggests that then, axonal connectivity is very important at the very least in constraining the shape of functional connectivity. And as part of this, we can also find other interesting features, such as the fact, for example, that there are some nodes that are very important in keeping together, in orchestrating the communication between these communities. And when you map these nodes, lo and behold, what you can see here is that the nuclei that are very important in acting as an effective facilitators of intermodule communication are indeed, dopamine, serotonin, noradrenaline. So this, ascending neurotransmitter system that are actually wired, here we're talking about axonal connectivity, as between network connectors. So why did I take this digression, perhaps a little technical? If we can quantify this process, so based on what I show you here, what I shown just a while ago, and this is being replicated in humans as well. Many people are led to think then, based on this, this corresponding functional connectivity equals to structural connectivity. And if you read a lot of clinical paper, many people say, oh yeah, that's this, like lower connection density, higher based on functional connectivity. What I would like to tell you in the next 3 minutes, I guess, or 5, is that this is simplistic. This is, in fact, wrong. Structural connectivity constrains, so shapes functional connectivity, but these two terms are not equivalent. How can I tell you? How can I make this statement? I'm going to show you 2 very short examples. One, let's not forget that the brain is a dynamic system. And when you measure functional connectivity, you tend to integrate brain activity over a large window, let's say 10 minutes or so, ignoring these up and down. And so what we did, I'm going to show you in the mouse again, one simple and perhaps, strong demonstration that is not the case. Functional connectivity is embedded in different things. So this brain activity is fluctuating. And this research, it is a bit technical, but it is very easy to understand, was motivated by some findings that were reported by a number of labs, such as the fact, I'm sure you come across this research, that there are millions of calcium activity in the cortex of the mouse, as well as primates, wherever you can measure it. So spontaneous neural activity is got sort of a measurable pattern. And so, in the lab, we were wondering, but what about functional connectivity? Functional connectivity is in the resting brain, so we should have some sort of wave-like activity that we can detect with fMRI. Well, what I'm showing you here, what looks very noisy, is just the signal in these 3 regions, blue and red. And you can see, the ups and downs. And when I just show you, this is raw signal in a single mouse light, you can see that in fact, you can by eye detect some waves. This is the cingulate cortex, activity is high, this is down, fine. But look now, the activity here with respect to the neighboring areas. So red and blue, this is blue and red for the switch. But look at that, here we have red here and red here. So then we can and now the question is, how do can you just, so this is called coactivation waves. How can we just map this? Very, very, very easily. You take an fMRI time series. This can be done in humans as well. We are doing it in humans. You take an fMRI time series and you find these recurring patterns of wave-like activity. Basically, you use the individual frames and you see where they are similar. And by doing so, you can create these maps here, which we call recurring fMRI states, or CAPs. CAPs stand for co-activation pattern. Here, there is no correlation. This is just ups and downs of neural activity. And look at these patterns, how rich they are, and how they change. Blue means a trough, a bold activity. Red means a peak. And the most striking feature here, just keep in mind, if you use these ones, you can get a functional connectivity network by measuring correlation. But if you just deconstruct them into their constituent patterns, that's what you get. And what is really striking to us is this. These patterns are organized into opposing networks, which means that they go up and down, up and down. And this is very nicely or more effectively captured in this figure, in these videos here, which I hope I can activate somewhere. Yes, so you can activate and you can see really that now this is a bold time series, which you can transform into like a okay. So now, why is this exciting? It's because we can now move from this idea of functional connectivity as a like proxy from connections, which is misleading, to this. This is like a functional connectivity study where we can, this is like a mouse model of autism, I'm not covering this, but as you can see here, we can now see, we can translate functional connectivity aberrations into altered neural dynamics in specific regions. So we can see the activity is going up, red is the mutant where it shouldn't go, or it goes more down where it should go down a little bit. So we can endow dynamics, really neural dynamics, you can endow neural dynamics mapping, and that's where we want to go. So I hope this is enough to show you that functional connectivity is constrained by the axonal connectome, but it's not equivalent to that. Okay, there's more to it. If you're not convinced yet, I'm just really touching upon one slide and then moving to autism. There are also traditional approaches we can use in the mouse, and we've been doing that. So one way to do this, for example, is silencing a node. This we can do. Please consider this as a bit of a teaser, because I'm not telling you the whole story. But long story short, if I just silence chronically, okay, in the mouse, the prefrontal cortex, so a hub region, instead of seeing lower functional connectivity, I see paradoxically increased functional connectivity, which is, I think, a nice demonstration. The ERC is replicated as part of a bigger study, which I cannot disclose now. We have a mechanism for this. So if you're interested in this, get back to me. I can tell you what we found. But so that also causal perturbation demonstrate that functional connectivity is not a monotonic correlate with axonal connectivity. This is a nice demonstration. I'll try to show it later. And as mentioned, if you're interested in this sort of research, stay tuned and watch this space, so to speak, because we've been lucky enough to get funding for a series of manipulations, and we will be releasing the data hopefully very soon on the first round of studies. So please keep this in mind. Functional connectivity is constrained by the connector, but it is a functional measure. Now, 9 minutes or so, just to tell you what we can make of this when we try to understand autism. I won't spend time to introduce autism. I'm sure you guys know. The only message I would like to tell you is this. Heterogeneity is a common dominant theme in autism, and in fact, it is a spectrum of disorders. You know very well. The second message is autism is strongly heritable. In fact, to my knowledge, is the strongest heritable complex disorders of the brain. And it is strongly genetic. However, there are so many genes that have been associated with an increasing number. And none of this explains more than 1 or 2% of cases. So that's a very big needle, right, to shift the. And so all the people in autism are trying to make sense of this heterogeneity. included, okay? Now, why is this important? This is important because of this. Well, people working in autism for many years trying to find a level of convergence because, okay, that's in terms of etiologies, there is so much heterogeneity. So many clinical scientists says, okay, fine. So the basic etiology is heterogeneous. There must be then a level of convergence because in terms of symptoms, yeah, there's heterogeneity, but there are some also discourse symptoms. So where is this convergence happening? And you know, and the obvious candidate goes at the level of surface, right? So what clinicians have been doing is, okay, let's select patients, let's stick them into the scanner, and let's calculate their functional fingerprint in order to find actually a single signature of brain dysfunction associated with autism. These are some of the results I show you, and this is very exciting. However, results are all over the place. This is showing reduced connectivity in autism, increased connectivity, and even Mosaic now recently increased and decreased. So, you know, they don't make sense to the results. So why is that? I am an imager, so I tell you what people have been discussing now in this field. There are two leading hypotheses. One is the so-called reproducibility crisis. You know, technical things, scanner inconsistencies, people have been trying to rush and publish, and so that's why there's such a mess. The second question, the possibility is the following, the one that's what I would like to propose. Perhaps this inconsistency is that there isn't a single signature of autism, but there are many. And if you just add them kind of randomly because you pick patients without knowing them, you end up with this. How can you test these two? How can you tell apart these two hypotheses? Ooh, I'm a little late. Well, one way to do this is the following. You could technically select syndromic mutations in patients and map for each of these mutations the connectivity pattern. This is being done, but it is also very slow and difficult. Why? Because these mutations are very rare. In order to get 20 patients, you need to do it like Ireland or Europe. Okay? So let's talk about the what we have in the lab, we say, why don't we just accelerate research? We can recreate some sort of a spectrum by mapping many mutants in the mouse with fMRI, and then try to translate this, you know, with fMRI to humans, so to speak. So we recreate a spectrum, and the idea is the mouse can control very well a number of things. You know, you have like their own controls and so on and so forth. And in the process, you can also maybe understand some of the mechanisms. And that's what we did. And we did this because we were prompted by the results of this study, which I'm going to show you very briefly because I don't have enough time. This is one of the examples where you select patients with a single mutation. This one is called 16p11. It is a copy number variation from the Simons Foundation. They gave us the data. These patients here have lower connectivity in the prefrontal cortex. We replicated the measurement in a mouse model, and we have exactly the same thing, lower connectivity in the prefrontal cortex. Very encouraging, because these changes can be translated, as you will, across species. We also have an idea about the mechanism, but I want to skip this, because I want to make a point, a stronger point later. I want to tell you another story and save this 5 minutes. Let me get back to the original question. So why such heterogeneity in humans? Is it like because we cannot image consistently things across changes, across sample size, or is it because indeed there is no such a signature of this connectivity in autism? Now, as mentioned, what we did, this was really a heroic effort. We mapped all these mutations in the lab over the years. As you can imagine. But when we put this together, and this is still in release, but we are working on this to write the manuscript, what we did is, first of all, seemingly unrelated mutations like these four, because as far as we know, they're not related mechanistically, they can converge to produce similar patterns of functional disconnectivity and aberrations. Blue means lower connectivity, and we have a lot of heteromodal cortices here, for example, prefrontal, so regions important for social brain. So nice. However, when we zoom out, we begin to add more mutations. We begin to see that we have a shift from prefrontal to more motor sensory. We have even a mutation where there is no change in the cortex. I'm just showing the cortex, and that's the most exciting bit. We go from blue to red. We have mutations that produce, instead of reduced connectivity, over connectivity. So I think this, in my opinion, is pretty clear. in that, it is very unlikely to expect that there is only a single fingerprint of connectivity that is associated with autism, because these two, if you put these two guys together with these two, the sum is zero. Some are hyper-connected, some are hypo, okay? And in the clinic, you are randomly sampling across this. And this is not only in my lab, because we collaborated with Verona, very talented. And we said, let's put together our mice. We did it. We have 18 mutations. We still have 3 clusters. One is hyper, one is hypo, one is somewhere in the middle. So I think it is a very clear demonstration that there is no single signature, but however, we can cluster these mutations into groups, into signatures. And that can be interesting. And I want to end on this bright note. I want to show you in 5 minutes. Okay, probably I'm going to overflow for 3 minutes, but this is very simple story that I want to throw at you somewhat in order to analyze on a brighter note. Because I think we can make use of these signatures to powerful heterogeneity in the clinic. And I want to show you the first. It's very short, very clear in my opinion, but let me know, let me have your feedback before this is unpublished. Okay, so this is the only difficult thing. So in autism post-mortem, there's this evidence that children with autism are supposed with purpose to have an increased density of dendritic spines. This has been replicated by 3 groups. And that's this big study, very elegantly recommended to everybody in my lab, from the David Salter lab, they're showing that the post-mortem samples with higher density of spines have also biochemical evidence of mTOR pathway hyperactivity, which I'm familiar with what it is, but I want to explain what it is. It's not important. What is really important is this, that in this pattern of hyperactivity, this biochemical pattern that is being very well characterized, there are a number of autism genes that are converging on this pathway. Okay, many of them, let's say at the very least 10. Okay, different levels, really very close, some just more distant. So this is a major pathway, and in human post-mortems, this has been related to the increased dendritic spine connectivity. So what we said, okay, interesting. Can we just recapitulate this pattern in the animal and measure functional connectivity? So in other words, what is the connectivity fingerprint that we get when we have hyperactivity of this? Whatever this is not important, it's an example. Well, okay, let me just show you, because in 3 slides I'm done. First, this is a sanity check. When you just recapitulate mTOR hyperactivity in the mice, as you would expect, you have higher dendritic spine density, okay? Which is good, because that's where it all started, okay? Nice. What about connectivity? Well, connectivity is this. So when we have more spines, you have like a strong hypersynchronization of the brain. So now we know that for this, cellular level alteration, you have this fingerprint. You have more synchronization, aberrant synchronization, over-synchronization. So far, so good. Now the question, this was another sanity check. Okay, are we really with the model here, are we really producing hyperactivity to mTOR? How can we make sure? Well, we can use a drug during development in the mouse we can do, which is called rapamycin, and reduce mTOR activity to see whether we can rescue the pattern. And that's what we did, and that's what we achieved. Patterns, it totally rescue. This is the hyperconnectivity I showed you before. This is the rescue effect. You see that blue is the negative of red, so as good as it gets. What is even more interesting is that we can rescue dendritic spine density, nice. And when we do that, we have a total abolition of this overconnectivity. So this shows that our mechanistic idea is very well modeled here. We also have behavioral changes. And what we found is that this hyperconnectivity is a good predictor of stereotypies that these mice have. These mice have motor stereotypies, reminiscent of some autism symptoms, which are totally rescued also by these mice in treatment. So it's nice because we know that these cortico striatal circuits it is aberrantly working in this mice. And that's the mouse part. I have two more slides, probably three tops, to show you how to try and translate this in humans. Okay, so here what it is coming a little subtle, but I think it's very easy to understand. So our hypothesis is the following: we have so many human databases with resting state fMRI in people with autism. This database is called the MIND. There are hundreds, literally hundreds of kids that have been scanned. This is idiopathic autism, so not mTOR hyperactivity or other kind of autism. You have the data there. And our hypothesis is a simple one. If this change is so prominent, there are so many genes converging on this pathway, there must be a signature of hyperconnectivity in these kids. There must be, because there are so many genes. Maybe it's not the prominent, but we should be able to capture it. How do we do it? Well, the way we did it, we used like some sort, let's call it machine learning. It's a very simple approach, which basically we looked for a signature of hyperconnectivity in this database, okay? And as a matter of fact, we found it. We found it because across all these sites, you see that there are areas that are hyperconnected in these kids. And when we clean for size, we use all multivariate regression, you name it, we control for everything, IQ, age, whatever. What is surviving very nicely is this bilateral foci of hyperconnectivity in the anterior insula, which is the one that is usually found in the mouse as well. Nice, so that's giving us clues we need to look for. Then what we did is this. We can see what network is affected, because the insula is a hotspot where things are happening, and what network is related there. So in the human, you see that this network that is hyperconnected is people with frontal striatum, which is exactly what we had in the mouse, so encouraging. And what we also found is that the kids that have this hyperconnectivity in these areas also have stronger stereotypies, motor stereotypies, which is another interesting thing, because that's exactly what we found in the mouse. However, what is linked here is the molecular link, right? So how can you tell what this is the coincidence? Well, there isn't much I can do in terms of molecules. I can just go on genetics, or I can just go and say, not to go to this kid and say, give me the blood, because also these data are anonymized. But what we could do is the following. We could measure the interactome at core, and what we found is that the mTOR proteome interactome is spatially correlated with expression of this. This pattern here is spatially correlated with expression pattern of heterogeneous interactome. How can we do it? By using the Allen Brain Institute postmortem human brain gene expression analysis. That you can do, that is available. Last slide, and then I'm going to stop. Okay, so this is not causal demonstration, but this is going towards the right direction. So one last thing that was bothering me, okay, this is interesting, but this is for the whole population, which doesn't make sense. Autism is not only mTOR, autism is many other things. So this must be driven by a cluster of patients, if you want to believe this. And as a matter of fact, we did find a cluster of patients. We used another machine learning, very simple decomposition, and we found that we can divide the connectivity profiling of all the patients. Every line is a patient of this database, and we found that there's this subtype here in which this enrichment is much, much, much stronger. And if you remove this patient here, the enrichment is going. The enrichment in terms of interactome fMRI. So we believe that in this way, we can go and fish out, pick out patients that have, functional characteristics that can be driven by biologically informed studies in the mouse. That's where I'm going to stop. I know I went a little 5 minutes further than I was hoping to. So take-home messages. Well, I think I said it very well. That fMRI can be used to deconstruct the signals. Remember that we can try, that's what we try to do. Deconstruct and understand what really connectivity. We can use these as a way to reconstruct also all things that the process of heterogeneity. These are all the people that did the job. I didn't do anything. Just work, and I was lucky enough to get a couple of friends, to be honest. And I don't know what else is doing, but these guys are really great, and I'm so grateful to them. And I have, of course, to collaborators, in my lab all over the world, and all the funding agencies that we were lucky to get engaged here. And thank you very much for your attention.
so excited. I'm looking forward to the first results together on the robotics project and on fragile. So now, the talk will go into discussion. If you have any questions, please ask. Also, after we have some coffee and tea outside, we'll stay there anyway. So if you have other questions and also questions about Ulysses, you can discuss after. But please, go ahead.
Just a quick question, a technical question. How important is the anesthetic effect in interpreting your data? Yeah, thank you for bringing this up. So, sedation. So the short answer to your question is that it's very important. So it is very important to control the physiological state of the animal. We don't use anesthesia, we use sedation. But overall, I think that the better way to put this question is, how can you achieve real quiet wakefulness in the mouse, in a non-compliant species? That's the subject of discussion. Obviously, deep anesthesia is introducing this wave, like hypersynchronous waves, up and down states, and then eventually you're going to see this one single network within the whole cortex, which is super synchronous. So you have to steer clear of that. With sedation, what we see, well, you can see yourself. When we just compare the axonal connectivity with functional connectivity under sedation, you have like a one-to-one correspondence. So this means that we have enough richness. I tell you also that now we have up and running awake imaging in the scanner. It's very challenging. And this is, roughly confirming what we've been seeing so far. So yes, anesthesia is potentially important, but now I think we have the tools and the means and like the interpretative also data set to guide us and to understand whether we are sedating the animal correctly. And this is not only mouse, also in primates, there's a lot of research that when sedation is low, you can preserve this structure. Just a quick follow-up question. You mentioned that possibly doing awake imaging. How long can you do that for?
In awake?
Yeah.
Okay. Now the problem with awake is, I want to be very honest. I'm an honest guy. The thing is that I was never really a big fan of awake in the mouse because I consider mice not to be compliant and intelligent enough to understand that that for half an hour keeping still. That would be good for them to get the reward. They would just, associate the reward to the good. They're not intelligent enough. But I did that because I kind of got tired of this review of questions about anesthesia. I say, okay, I want to get now another set of questions which they're going to bug me equally even more, which is, okay, what about stress? So that's another way of putting this. I know that the animals get stressed even if you habituate them. But overall, I think we managed to keep them pretty much still under mild restraint for half an hour. And then if you release them, for example, they are happy enough to come and eat food on your hand, which is good because when animals are very stressed, they don't eat, for example. You know very well everyone's very stressed. So I think I'm not denying that it's not stressful, but I'm just saying if it's possible to reach at the very least now, this is still unpublished and we're working for half an hour or so. Half an hour is good to get. Is good enough to get very, very high quality resting state. But that's still to be, it's work in progress. Sure.
Is there any other question? Well, I have one understanding. I think we will get some coffee outside. I'm very fascinated about the neurodynamic that you have shown. Just a comment from you. Do you believe we can actually apply this as a biomarker in drug discovery for pharmacological efficacy in neurodevelopmental disorders?
Oh, yes, definitely. Okay, so yes, the answer is by all means. We have already interesting data. So the interesting thing of dynamics is that it is, if properly done, it is way more sensitive than this steady state functional connectivity. There is, however, a big catch. There are a couple of unresolved problems with dynamics. Well, first of all, dynamics is, of course, adding another dimension. It is a very rich dimension. So there are big problems in the field, not related to our approach only, but it is, for example, when you map dynamic, the problem that we are still grappling with is how many states do you select? Because dynamics can be
It depends on how, you know, how fine-grained you want to go for. And so the big question is, there are, for example, let's suppose you want to compare two groups. One is like neurotypical development, the other one is autism. Okay, neurotypical development, you have a big group and you identify, I don't know, eight states that explain, say, 90% of the variance. Cool. All the rest you can ignore. You say they're there, but they explain very little variance. What we don't know yet, and we are working humans as well, and that's where we are kind of not stranded, but scratching our heads, is what about autism? Do you expect the states to be the same, or do they change? Do they have like a set of new states? Is the brain like undergoing configuration? And that's not a philosophical question. This is a very practical question, because if we assume that they are the same, you can use some quantitative measurements and compare them head to head. If there are new states, those are by definitions uncomparable. It is interesting per se, because that's exactly maybe the biomarker thing. Okay, there are new states. For example, when we do proof of concept studies with drugs, we use like a cholinergic stimulant, we try. We can tell very clearly that the states, as you would expect, they entirely. So you just have, you go from a set of 6 states that look like something to 6 other states that look completely different, which is okay, because you know you are. So the answer is yes. How to do that reliably? It is something that many groups are working, and you know, I have like a couple of people working on this. That's what I'll be able to get back to you. But absolutely, and that is much more sensitive than functional connectivity, but like an order of magnitude in terms of statistical power. This I can tell you, unpublished, but probably soon we'll report something.
So I say thanks again to Alessandro for this wonderful talk.
Thank you.
And, yeah, so the next UlyssesNeuro Talk will be, as already said, will be on the 28th of February. We will learn a little bit about Charcot-Marie Tooth, so I hope to see you again. And please enjoy some coffee and tea outside. And also, if you want to subscribe to our newsletter, the next one will be out in a couple of weeks. See your email address. There's a little notebook outline.